Good afternoon, Mishika. How are you doing? So they must have started chapter 8, right? As per your book, Patterns of Inheritance. So I think patterns of inheritance must have been started in your school. Genetics chapter that you were talking about. Okay. So let's begin that chapter 8, genetics patterns of inheritance, right? So we know that the penultimate father of genetics is Gregor Johann Mendel. There is only one name that comes in anyone's mind. Okay. So let's talk about Gregor Johann Mendel first. The father of genetics, not the modern genetics, the classical genetics. Modern genetics will be T.H. Morgan, Thomas Hunt Morgan. But the classical genetics and overall genetics, he is considered to be the father of genetics. So lived from 1822 to 1884. He was born in the European country of Austria. Okay, he was a monk, and he used to call the genes as factors because there were no discerning techniques at that point in time from 1980 to 2084 that could discern the genetic sequences or gene sequences. So he called them factors, okay, and then he experimented with Pisum sativum, that is the pea plants, Pisum sativum. I'll tell you all the reasons why Pisum sativum was chosen. Then he thought that heritable factors, which are also known as genes nowadays, so simply factors or heritable factors, which are known by the name genes nowadays, are retained. Okay, they are retained and they retain their individuality generation after generation. Okay, so principles of genetics was developed in the mid 19th century, and he experimented with the pea plants, okay, by crossing various strains and observing the characteristics of their offspring. So that is what Gregor Johann Mendel did. In overall, he studied 7 traits of Pisum sativum pea plants, and he was the first one to use the two very renowned terms known as, or they are referred to as, dominance and recessiveness. Okay, and as per him, the 7 characters that he chose in pea plants, on which he described or using which he described the entire basis of Mendelian inheritance, okay, were the following. Number one he chose was the flower color. D for dominant, R for recessive. So the dominant one was purple, recessive was white. Secondly, what he did was he chose the flower position of the flower. Dominant was axial, recessive was terminally placed, terminal. Okay. Number 3, stem length. So when it comes to stem length, the dominant one was tall, recessive one is dwarf. Okay. Number 4, seed shape. It's round is the dominant one, wrinkled is the recessive one. Okay. Then seed color. Yellow is the dominant one, green is the recessive one. Then number 6, pot shape. That is inflated in case of dominant, okay, constricted in case of recessive. Then 7 is the spot color. Green in case of dominant, okay, yellow in case of recessive. So these were the 7 characteristics which were chosen by Gregor Johann Mendel when he experimented with the specific Pisum sativum or pea plant. Okay. So these were the characteristics that he worked upon. Now coming on to why pea plant was chosen by Mendel. Okay. Why were pea plants chosen by Mendel? So what could be the answer to that very question? Why did he only choose pea plant? Why not any other plant? So that is again a very, very conceptual question pertaining to genetics, right? So the first thing, when it comes to specifically choosing the pea plant, was Mendel selected garden pea plant for his experiments because of the following reasons that I am going to write. Okay, so Mendel selected the garden pea plants because of the following reasons. Because point number 1, the flowers of this plant are bisexual, hermaphrodite. Okay, so both the sexes are here, so there is no chance of intermixing that it depends on the pollens from any other flower to be pollinated. So it is a bisexual flower. So the flowers of this plant are bisexual, so self-pollination will do the job. Okay, it doesn't depend on any other type of pollen grain. Okay, so this plant are bisexual. It doesn't depend on cross-pollination. Self-pollination will cause fertilization. And since they are self-pollinating, okay, so I can both go for self and cross pollination if I like, right? So then point number 3 would be the different physical characters that I had mentioned, the 7 different physical characters which was just mentioned are extremely easy to recognize, discern and study. Okay, the different physical characters are really easy to recognize or discern and study. They have recognizable traits which will not pose any kind of problem when it comes to making the analysis and interpretation after the experiment. If the traits are not recognizable or are very hard to recognize, very difficult to recognize, then what happens is the scientist can make errors. The probability of the error compliance or the error emanation becomes more and more. It becomes higher and higher. It shoots up. So that is what we want to avoid. There should be an easily discernible characteristic showing plant. Then one more point was they have a very short lifespan, and also they have shorter lifespan, okay, which will be time adequate, and also they are very easy to maintain, means are economically feasible. You won't need any extra media or very, you can say, exorbitant amount of nutrients, minerals and all in order to maintain this kind of plant. So that is what the thing is all about. Okay, that these are the reasons why Mendel chose the pea plant in the first place. Okay. Now if we come to several things that I'll be telling you here. The meaning of inheritance. Okay. First of all, let's come to the meaning of inheritance. So parents and offspring, okay, often share observable traits. You might have noticed. So Mendelian inheritance or Mendelian genetics or Mendelism is a set of primary tenets relating to the transmission of hereditary, you can say, characteristics, transmission of hereditary traits or hereditary characteristics from parent organisms to their offspring. Characteristics from parents to their offspring. That is, you are inheriting something, that is, you are inheriting the genes, the inheritable factors, right? Now, this entire way of transmission, the mode of transmission which will be expressed, which will not be expressed, which will be state dominant, which will be recessive and all, that is all defined by the term genetics. Okay, so the initial work derived from the experiments of Gregor Johann Mendel were published in, you can say, 1865 and 1866, which was rediscovered in 1900 and were quite controversial. You must be knowing they were quite controversial. Okay, so all the controversies, okay, were put to rest after they were tested again. Okay, but the units of inheritance that were known as factors, the hereditary factors, the units of inheritance are now known as alleles. That is the alternate form of a gene present on the homologous chromosome. That is the perfect definition of alleles. So alleles are alternate form of the gene present on homologous chromosomes. Now, what are alleles physically? So we know that humans have a chromosome count of 46. If you have 46 chromosomes, that cell is known as diploid in nature. 46 chromosomes. Okay. It's centromere which has CENP histones, that is known as diploid cell or 2N. If you have 23, it is known as haploid. Okay. Now, but how do we get to 20, you can say 46 chromosomes? So after meiosis, what we have is in germ cells, that is the cells present, or the sex cells we should be saying, that is the testis and ovary, they produce sperm and ovum / egg. So these are the only cells which are known as germ cells, and production of germ cells occur by the cellular division known as meiosis. Meiosis is also known as reductional division. Reductional division. So I'll give you an overview, even though meiosis has not been taught to you. I'll give you an overview so that you can understand what alleles are and how and why do we call each cell diploid or haploid. So what happens is we have something known as a cell cycle in case of eukaryotic organisms. Okay, cell cycle means the entire journey of the cell from it being a daughter cell to it being able to completely divide into again two daughter cells. Okay, so that is known as the cell cycle. The cell cycle has the following stages. I'll just simplify it for you. Okay, you have G0 stage, you have G1, S, G2, M. These are the many stages. This is the quiescent phase, means the cell is arrested. The nerve cells, the neurons, the cardiomyocytes, the skeletal muscle cells, these are all arrested in the G0 phase. This is known as quiescent phase. Means these cells cannot divide. Okay, quiescent phase of the cell cycle. Some nerve cells are arrested at G0 also. That's a different thing. So the neurons, they do not possess or they do not contain or comprise of centrioles, which will produce the kinetochore or microtubules necessary for cell division. So that is why neurons cannot divide. Again, one more reason. So the thing is, they are arrested at G0 phase. But the cells which are able to divide, like the follicles of the hair, the skin, the RBCs and all, so what happens is in order for a cell to divide, what you need to do? You need to double the organelles also, because if you divide it just like that, organelles may not be equally distributed. Okay, you have to make two nucleus, you have to make two mitochondria if there are one, right? You have to double the endoplasmic reticulum, Golgi apparatus, lysosomes, okay, then peroxisomes, then vacuoles. So you have to double the organelles also. So organelle doubling mostly happens in the G1 phase. In the S phase, you need to double the DNA also, DNA duplication or DNA replication, because 46 chromosomes are present. You need to divide it into two cells, so both must contain 46. So you need to make the DNA doubled. Okay, what happens is, suppose the chromosome number remains constant, DNA number becomes doubled. So you have 46 chromosomes. So I am drawing only 4 here. Suppose 4 are equal to 46. Okay, so after the S phase, what will happen? Sister chromatids will be developed. Okay, this will be used as a template and new DNA strand will be formed complementary to it. Okay, and each chromosome, if I am drawing like one, don't think it as single stranded. This contains double stranded DNA. Okay, that's the concept. So when a new chromosome is formed like this, it means it is also containing a double stranded DNA. It is containing a double stranded DNA like this, but this is a simplified image. So here what happened? In the beginning, if I again compare, before S phase you had 46 chromosomes. After S phase also you will be having 46 chromosomes only. But each chromosome has got its sister chromatid. Okay. You have sister chromatids for each chromosome, meaning that DNA here is 46 double stranded DNA. Here it has become double. It has become 92 double stranded DNA. DNA number has become doubled, chromosome number stays constant, but the thing is genetic material has doubled. Okay, that is the way genetic material has doubled. Now, when we talk about, you can say mitosis means now you have 92 DNA. What will happen is each will be divided like this. Okay, each will be divided like this, and you will get two cells of, you can say 46 chromosome, 46 DNA. So like this, from one cell you get two cells which are also diploid. So the parent cell was diploid 2n, S phase G2, then M. In the G2 phase DNA repair and all will occur, okay, which I'll explain to you when the cell cycle chapter starts. Then we enter the M phase. M phase is the division phase. In the division phase, you will have 2n, one daughter cell, another daughter cell, again 2n. This is simply the mitosis. This one is mitosis. That is why it is also known as equational division. Okay, equational. Because the ploidy, it is diploid, this is also diploid. The ploidy has not changed. It remains equational division. Okay, that is mitosis. But in case of germ cells, by which we will be able to reproduce and transmit or transfer our character or trait or genes, the entire genetic material to our son or daughter, those undergo meiosis. So the path till here, the S phase is the same. You will be having double the amount of DNA. Okay, and 46 chromosomes. Till here, that is the S phase. Now, after this, when they will enter the G2 and then M phase, the meiosis will do what? In case of meiosis, it has two stages, meiosis 1, okay, and meiosis 2. In meiosis 1, what will happen is the homologous chromosomes, okay, they pair up. Okay, they pair up means what happened was, in the sperm cell, now go backward to a sperm cell. In the sperm cell, there are only 23 chromosomes. Okay, in the ovum there are only 23 chromosomes. Now when they meet, and suppose this is the zygote, okay, this is the zygote, suppose. Now they have 46 chromosomes. Okay, now let's make this a zygote that has undergone S phase. So let's make this the zygote. Okay. Now they have 46 chromosomes. This is 2n, this is n, this is n. Okay. Now what happens is, the girl and a boy, they are completely similar except for the sexual characteristics. Otherwise, you have heart, I have heart, you have blood vessels, kidney, liver, I have all those organs, right? Eye to ear to nose, everything is same except the sexual organs and secondary sexual characteristics. That is controlled by the last chromosome known as X or Y. That is the allosome. The rest of the chromosomes, the non-sex chromosomes, are known as autosomes. Okay. So that is autosome and allosome. Now when we begin the discussion, okay, what happens is, suppose this is chromosome number 1, 2, like this will be having till chromosome number 23 here. This is also chromosome number 1, 2, till that we will be having chromosome number 23 here. So when we have 46 chromosomes, so this is chromosome number 1 from this cell, this is sperm, this is ovum, okay. This is chromosome number 1 from this cell, that is sperm 1, ovum 1, or I can say paternal 1, or from father, maternal 1. Same 2, 2. So chronologically same. So sperm chromosome number 1 from the paternal side and chromosome number 1 from the maternal side. Okay. Unless you have this 23rd pair, this 23rd, suppose here is the Y chromosome, this one is the X chromosome. So we'll get XY here, okay, X and Y here. Okay. So, wait, X and Y here, yeah. So this will be X, this will be Y, okay, Y and X. So unless we go to chromosome number 23, all other chromosomes are homologous. Homologous means, the meaning of homologous is their DNA sequence is not completely identical but are similar in the sense that they code for the same proteins, they have the same set of genes, because the mother and the father are completely same except for their sexes, and sexes are controlled by the last chromosome 23rd. Okay, that will not be same. This will not be called as homologous unless it is X and X. Okay, X and Y are never homologous. Okay, but all the 22 pairs, the autosomal pair, this is the allosome. This pair is the allosome. All the other 22 pairs are known as autosomes. Will have the same genes coding for the same proteins, because chromosome number 1, now let's get into it. Suppose chromosome number 1 from zygote, this is chromosome number 1, chromosome number 2 zygote, sorry, this is sperm. Okay. This is a sperm 1, 2, this is an ovum. Now, fertilization has happened in the ampulla region of the fallopian tube or oviduct. Now you have produced, you can say, a diploid cell, 46 chromosomes. Okay, so this is one from paternal, one from maternal, means sperm, ovum. This is again 2 from paternal, 2 from maternal, that is again sperm and ovum. So if the first chromosome, so let's suppose it contains the gene for human growth hormone, which is produced by the, you can say, pituitary gland, the anterior pituitary gland or adenohypophysis gland in the brain. It will take care of our height, it will cause our height to grow, it will make us taller, right? It is responsible for bone and muscle growth. Same way, chromosome number 1 here will also contain HGH because the chronological order is the same. Now in the zygote, this one is the zygote which will become fetus and eventually the baby. So chromosome number 1 has got HGH. Here also it has got HGH. Now suppose the father is 6.6 feet, 6 feet 6 inches, that is 6.5 feet tall. Mother is 5 feet 2 inches tall only. Okay. What will be the height of the children? Which one will it be bestowed with? Will it be tall height or will it be a short heighted mother or father? So depending upon that means that the two HGHs are not same in their genetic composition, in their gene sequence. They are coding for HGH, but they have single nucleotide polymorphism or variation that we are talking about in evolution. Now let's zoom it in. Okay. If you take chromosome number 1, okay, from the sperm, HGH, this is from the parental side sperm, this is from the ovum or maternal side chromosome number 1 only. Okay. Now if I zoom into only HGH, okay, the sequence of suppose HGH from the father or parental or sperm is this one. ATG. I am giving an arbitrary sequence, mentioning an arbitrary sequence, assuming a sequence, that's all. It could be any sequence, or you can, if you want the real sequence, you can go to ncbi.com, okay, and get the sequence from there only. ncbi.com/pubmed. Then you can then search for any sequences also here. You will get the sequences of all the genes present. Okay, so I am taking an arbitrary sequence. You don't need to remember any sequence here. Just this is the conceptual understanding. Okay, suppose this is the gene coding for HGH in the father who is 6.5 feet tall. Now this is the HGH gene produced by the mother who is 5 feet 2 inches. Okay, 5 feet 2 inches tall. Now what is the difference? Suppose this is the difference. What are the differences? On the second position you have T here, you have C here. Fourth position you have C here, you have T here. Rest all are same. So these are the two single nucleotide polymorphisms, two variations, right, which will cause the protein, because genes are coding for proteins. Human growth hormone is a protein hormone. Okay, so the thing is, this protein HGH and HGH1 and HGH2, suppose this is HGH1, this is HGH2 made by the mother, they are different in their structure because proteins have quaternary shape or quaternary conformation. So it depends on each and every amino acid on each and every position. So later on when I will be teaching you the molecular biology, genetic code, you will be knowing that you should divide the, you can say, the number of nucleotides or bases present in the DNA sequence into group of 3. Group of 3 nucleotides are known as codons. This is known as codon, and each codon codes for one amino acid. That is how proteins are produced from DNA. That is how the DNA determines the sequence of the proteins. So ATG suppose codes for glycine amino acid, ACG suppose codes for leucine. So didn't you change the amino acid here on the first position? So now because of the change in the amino acid, here suppose it is coding for serine, this TTA is coding for alanine. So again amino acid changed. So because of the change in the amino acid, this is just a supposition, okay, to just show you the concept. I'm not teaching genetic code here, okay. So because that is not to be mucked up, ATG may code for something else, I've just taken this as an assumption, okay. So the thing is, if amino acids are changed, the protein structure and conformation will differ, and then you know that if the conformation is less favorable, is not conducive, it may not bind to the receptors, HGH receptors on the muscle and bone and may not cause them to grow rapidly. And if this is a very high quality HGH, this is little bit low quality, so this one will have proper conformation. Okay, proper conformation means proper structure. The structure will be very, very proper, appropriate, which will bind to the receptors present on the bone and muscles and make them grow better, make them grow at an accelerated rate compared to if it was the case with, or if it was the case pertaining to HGH type 2, which is coded by the mother. That was is 5 feet 2 inches tall only, and this guy is 6.5, you can say, feet tall. So this is the meaning of, you can say, alleles. So they are both coding for HGH, but they are the alternate forms of the alleles because they are not completely identical. Their sequence differs a little bit due to some single nucleotide polymorphism. Polymorphism means variation. It is polymorphic, it is not same. And they must be present on homologous chromosomes, because if it is chromosome number 1 and chromosome number 2, if I compare, I can never compare the same genes, because chromosome number 1 is carrying a HGH. Chromosome number 2 will never carry HGH. So it will never be the alternative form of the same gene. It is suppose coding for insulin. Okay. So you cannot compare insulin and HGH, right? You can compare only alternate form of the same gene. So same gene is present on the same chromosome number, chronological chromosome number, and these same chromosome numbers are known as homologous chromosomes, because the traits are same in parent in both the sexes except the sexual characteristics. So the same chromosome number in both the sexes, they contain the same genes at the same specific location also. That is known as locus. The plural of that is loci. Means if you are talking about a submetacentric chromosome, then suppose you have let's suppose this is how we draw a chromosome. Centrome here. So suppose submetacentric, okay, this is the short arm. So suppose a short arm of chromosome number 1 contains HGH gene. So at the same location here on the short arm of chromosome number 1 from parental or father or sperm, this is from mother or ovum, at the same position of chromosome number 1 will be HGH. That is same locus. Okay, the short arm is known as P arm. Okay, P means petite, long is Q arm. Okay, P and Q arm, they are called as for the chromosome. P arm of chromosome. So you may write like 1P. 1P means chromosome number 1, short arm. 1Q means chromosome number 1, the long arm. Okay, that is how it is denoted. So the location will be same. It wouldn't be like HGH is located here. It wouldn't be. This is wrong. This will be the same. So alleles are located on the same locus of the homologous pair or on the homologous chromosomes, but are not identical in their genetic sequences. That is why they are known as the alternate form of the same gene present on homologous pair of chromosomes or homologous chromosomes. Okay, that is the way alleles work, and that is the concept of alleles and how they are different and how they are known as alternative form of the same gene. So now, Ishika, do you have any doubt in allele? What are alleles? What is homologous chromosome? Okay, and what is variation? Why two alleles are called as alternative of the same gene? Why are homologous chromosomes required to complete the definition? Any problem here? Any query? Any doubts? This is a very, very conceptual topic. Okay, I want you to have complete command over it. Any queries, Ishika? Okay. So then we can proceed. Now you will talk about dominant and recessive. The dominant allele, as per the definition given in the book, they say that the dominant allele masks the expression of the recessive allele. That is the function of dominant allele. Then they say, as per another definition, that the expression of the allele that is matched, okay, that is matched, is known as the recessive allele. Okay, but does it really match the expression? As per the definition, it is fine. But what about conceptually? Does it really match the expression? Masking doesn't happen. What happens is, it is not a bodybuilding competition or a strength competition. What happens, suppose in this chromosome, you have the father HGH, okay? Chromosome number 1, father, or you can say sperm HGH. Again here, number 2 from mother or ovum HGH. Okay, these two are alleles. Now, this is a zygote. It has got one chromosome number 1 from mother, one chromosome number 1, okay, from mother, chromosome number 1 from father. Now, what will be the height of the child? Okay, whether girl or boy doesn't matter, but what will be the height? Now, coming into or thinking about this thing, if this is the condition, means it has got both the different alleles, okay, then which will be the dominant allele? So dominant allele will be decided by the name, capital T. Historic reasons, dominant is capital, this is small for the height. Small t means recessive. Now, what makes dominant allele? Will it beat it up, the small allele? No. Both have DNA sequences. They are not going to fight with each other, right? They are not going to mask the expression. What will really happen is, masking expression is a superficial definition, but what really happens is, suppose this one is coding for proper protein, okay, proper protein, or proper protein means proper hormone. This is a hormone, so it is able to bind to the HGH receptors present on bone and muscle and cause proper growth. But since this one is a dysfunctional protein, dysfunctional hormone, is not able to code for a good quality protein due to the variations, its conformation, its structure is not proper, so I can call it a dysfunctional hormone. So it doesn't bind, or binds very lightly, doesn't bind tightly, it does not bind properly to HGH receptors on bone and muscle. Okay, now you have two scenarios. Now, when the DNA is converted to mRNA and mRNA binds to the RER in the cytosol or cytoplasm, this protein is produced, but half the time this protein will also be produced, okay, but this protein will not cause growth. But when this protein is produced from this very gene, they will bind to the receptors and growth will be proper. Now, when growth will be proper, will you be able to discern or see or observe the effect of this allele? No, the effect of this allele, that is, the definition must be the effect of this allele is masked, is completely masked. Because when this specific protein coded by this specific allele binds to the receptors present on bone and muscle, HGH receptors, human growth hormone receptors, you see a specific type of growth. A specific type of growth means you see an appreciable amount of growth, okay, that makes the person tall, even if he is carrying the one of the homologous chromosomes out of the two, one of the chromosomes it is carrying the HGH recessive allele, still he will not be, or he or she will not be, a dwarf, because one copy of the chromosome contains or comprises of the favorable allele or the high quality allele, which will, when that allele specifically produces the protein. That protein is able to bind to the HGH receptors and cause the growth to occur. The body growth, the muscle growth and bone growth to occur. So that is why the effect of the recessive allele is not observed in the presence of the good quality allele or dominant allele. That is why it is called as dominant allele. It is masking the effect. It is not fighting that allele, it is masking the effect of the allele. Okay, that is what happens. So this is the concept regarding dominant and recessive. Now, there are something known as homozygous and heterozygous. Homo means same, zygous means allele. So the example that I was giving you till now was of heterozygous condition. Okay, HGH low quality or small t from the, you can say, mother, HGH high quality from the father. Okay, sperm, ovum. Okay, this is the zygote. So this is a heterozygous conditions where both the alleles are not identical in their genetic sequence. Okay, that is known as the heterozygous condition. Second term it capital T, small t. This is the recessive one, small t, low quality, high quality, simply capital T. So capital T, small t is a heterozygous combination. But if both identical alleles are paired together by mere probability in the zygote, because it is a genetics is a chance thing, it's a thing which is governed by probability. So suppose you get this thing, okay, capital T means both the alleles have high quality HGH coming from father, coming from mother. This will be known as a homozygous state, and this is known as homozygous dominant. So under no conditions will it become dwarf, it will always be tall. If you have this condition, small t means both the genes HGH is of the low quality. Okay, now there is no capital T HGH or high quality HGH to mask the effect, means both the proteins, this HGH protein also, this HGH protein also, both cannot bind to the receptors present on bones and muscles. So the growth will be very, very nominal, very, very less. So the person phenotypically will be dwarf. This is again known as homozygous condition, but homozygous recessive. Homozygous recessive. Only case where the homozygosity doesn't lead to the alternate form of the same allele or same allele composition to be united is the XY chromosome or allosome. Okay, because X, this is suppose Y chromosome, this is X chromosome. So the genes will be different completely because this is X, this is Y. Genes will not be same, but they pair up. That's the last pair. So they are known as hemizygous, means half of the genes. Hemizygous, they are the hemizygous pair because they are not homologous pair of chromosomes. Okay, whatsoever. They are completely different chromosomes. They are the allosomes, the sex chromosomes. They are known as hemizygous. Only in case of boys or males they are hemizygous. In case of girls there will be 2X, again homozygous. Okay, X and X will be again homozygous. So in case of girls it will be homozygous. Okay, depending on homozygous or heterozygous, but it will be homozygous, sorry, not homozygous, it will be a homologous pair of chromosome. Homologous pair of chromosome. But the genes could be that it could differ. Suppose in case of SARS-CoV-2 you have AH2 receptor gene present on X chromosome. So this may be a high class receptor, this may be a low class receptor. Okay, depending on the difference again. But chromosomes will be homologous, this will not be homologous, they are hemizygous, means completely different altogether. Okay, this is the concept regarding the hemizygosity. Okay, So if you are having hemizygous chromosomes, you can never think of dominant and recessive because they contain totally different genes. Okay, here you can think of dominant and recessive, dominant or recessiveness. Okay, so next up is genotype versus phenotype. Okay, so genotype, if I say genotype, means it refers to the alleles an individual, you can say, receives at the event of fertilization or the time of fertilization. You can say refers to alleles an individual has received at the time of fertilization. Then phenotype. Phenotype is nothing but this is the physical appearance of the individual, the characteristics that you are able to discern or see or observe. That will become better after an example. Okay, physical appearance of an individual. So let's do a cross now, known as the Punnett square. Very simple cross. Let's do a monohybrid cross. Mono means one character, hybrid means you are crossing, means you are going for fertilization. That is the meaning of mono. You will see the probability or chance events. Okay. So let's talk about a monohybrid cross. Monohybrid cross means, suppose I am taking the tallness as only one character I will take into consideration here. So I am taking tallness, okay. One side I have taken TT, one side small t small t. So this is homozygous dominant, homozygous recessive. The homozygous conditions are also known as true breed conditions in genetics, okay. True breed. They are also known as true breeding conditions. Always remember. Okay, they're also known as true breeding plants will make the or will contain the homozygous state alleles in either dominant or recessive form. Okay, now this is known as F0 generation, where F stands for filial. Okay, F stands for filial here. Okay, now what do you mean by filial? Okay, filial means son. Okay, so that thing means filial means son. Okay, so Mendel called it filial. Okay, what are the siblings that you are, what are the progeny that you are going to get? Okay, that is what he did. So let's talk about son or daughter, both you can call filial. Okay, the offspring or progeny only. In simple terms, offspring or progeny. So now let's cross. Let's take probability. So as per the, you can say crossing, as per the Punnett square, what I can do is I can write T here. This is one parent. This is another parent, small t, small t. Okay. What all I'll get? I'll get small t, you mix with this one, small t, capital T, small t, capital T, small t, capital T, small t. So all the progenies pertaining to the next generation, which is known as the F1 generation, will contain heterozygous state, but will be tall. Okay, that is the meaning of genotype. Genotype means, now see, the genotype here was it had identical genes. It also had identical genes, identical alleles, because it was homozygous. So this one was dwarf. This one was tall, but in the F1 generation, all are tall. But can you tell just by looking at their appearance what is their genotype? They are all heterozygous, but all are tall. Okay, you cannot look at the individual trait from outside the appearance of an individual or plant or an animal and discern or decide the genotype of that specific plant or animal as per that trait that you are visualizing or observing. So that is the thing here. It is known as genotype, means what all alleles does it have. If it contains capital T small t, it is a genotype. But phenotypically, all look tall. Okay, all look tall. So genotype will differ. This is a different genotype, this is a different genotype, because here it has got two same alleles, identical alleles. Here they have got different alleles. But phenotypically, this will also be tall, this one will also be tall. That is the difference between phenotype and genotype. Okay. Now when you cross, you go for selfing. Selfing means crossing in the same generation with the same type of individual, that is known as selfing. When you go for selfing, okay, in F1 generation, let's go for selfing in the F1 generation. So what you have got in F1 is capital T small t. This is what you have got in F1 generation. All were capital T small t. Now since all were capital T small t, so capital T small t will be served with capital T small t. Okay. Now what will you get? You will get a new Punnett square. Okay. Capital T small t, capital T small t, they start selfing like this. Capital T small t, here capital T, sorry, here it will be capital T, here capital T small t, here capital T small t, here small t, small t. So now you have got different types of genotypes. So if I consider genotypic ratio, so the trick that I'm going to tell you is check the number of repetitions of the same type of allelic combination. Okay, what you have to check? Number of repetitions of the same type of allelic combination. That is the trick to be known. Now what does that mean? Capital T is one type of allelic combination. How many times has it been repeated? Only one, right? Others are small t, capital T. So only one time it has been repeated here. Okay. So genotypic ratio, I can write it as 1 ratio. Now capital T small t, how many times has it been repeated? 2 times. You can see 2 times it has been repeated. 1 ratio 2 ratio. Small t, how many times has it been repeated? Again 1. 1 ratio 2 is your 2 ratio 1 is your genotypic ratio for a monohybrid cross. Okay, that is the genotypic ratio for your monohybrid cross. What you do is you just count the allelic combination repetitions. You just count the allelic combination repetition. That's all. In case if you want to find out the genotypic ratio. Now when it comes to finding out the phenotypic ratio, what we do is now you will look at the physical trait. For finding out genotypic ratio, I didn't ask you to see whether the person or the plant is tall or dwarf. Not a single time did I ask you. I just told you just look at the allelic combination and count its repetition. You shouldn't have to worry about the physical appearance, whether the plant is tall, dwarf, seed, wrinkled, yellow, nothing. Okay, round and wrinkled, nothing, or yellow, white, nothing like that. Okay, axial, terminal, not all those things. Just count the number of allele combination repetitions. Now when you come to the phenotypic ratio, okay, that time what we'll do is we'll look at the phenotype. Specific phenotypes means the physical appearance. Now capital T is what? Tall. So phenotypic ratio, if I write capital T is tall, capital T small t is again tall, capital T small t is again tall. So 3 are tall, only small t will be dwarf. So I can say phenotypic ratio is 3:1 because 3 are tall, 1 is dwarf. Okay, so that is how phenotypic ratio is calculated, by just looking at the physical appearance of the trait that we are considering or we have taken into consideration. That is the phenotypic ratio. Now, why was I calling it the chance, or you can say act of probability? Because once you have these two, okay, so you can produce a sperm which will contain only capital T chromosome. This is the father component. Now sperm will be produced, or sperm is half of the parent, 26, 46, it will be 23, either this one via meiosis or meiosis. The ovum will be produced, okay, from the mother with again this chances capital T, small t. Now during sexual intercourse, it is a mere probability that which two sperm and ovum will meet. If this and this meet, suppose 1, 2, 1, 2. If 1 and 1 meet, the kid will always be tall. If 1 and 2 meet, the kid will always be tall. If 2 and 1 meet, again kid will always be tall. But if 2 and 2 meet, if these two meet up, that is the 25% chance, very least, because 3:1 is 25% chance out of the 4 times, 1 time only, because you are meeting this 1 1, 1 2 and 2 1. They are making you tall. Only one event 2 2 mating is making you dwarf. So there is this 25% probability that if both parents are heterozygous in condition, okay, then 25% chance they can have that when it comes to tallness, they are heterozygous in condition. So the 25% chance could be that their progeny could end up being less tall or dwarf, okay, depending on the quality of the small t gene, okay. So this is the probability that I was talking about. This is the real world application of those crosses. Ishika, do you have any doubt? Okay, no issues. Okay then, thanks a lot. We'll continue.